Good morning, Year 12. So, this morning we're going to carry on with our natural selection topic, and we're going to talk about various different types of selection pressure. So the title for today is Selection Pressures, and we're going to start off with a starter here. So back your books, wherever you like to do your starters, 4 questions, pause me, have a go, it should take 2 minutes. Fine. So the main difference is between DNA and RNA. The main differences are that RNA is single-stranded, whereas DNA is double-stranded. DNA is longer, RNA is shorter. RNA contains uracil, DNA does not contain uracil, it contains thymine. DNA contains deoxyribose, RNA contains ribose. And I'm thinking that's probably everything. What does the term quaternary structure of proteins mean? It's where 2 different proteins interact, or 2 or more, to form a larger protein structure. Something like hemoglobin, where it's got 4 different proteins all attached together, and that's the way they interact with each other. So, where is genetic diversity increased in meiosis? Crossing over, independent segregation, and hopefully that's quite straightforward for people. And what's genetic diversity at population level? We're talking about the variety and number of different alleles, different types of alleles, I should be careful, found in a population. Fine. So today we're going to talk about, let's say, selection pressures. So we're going to talk about various different kinds of selection pressure, 3 different kinds. In actual fact, only 2 of them are on paper 1, but it doesn't seem to make sense to me to leave out the 3rd one. So we're going to talk about the 3rd one as well. That will be on paper 2. You're actually probably going to go back over that in year 13 as well. We'll touch on it today very briefly. The 2 we do need to know about for paper 1 are directional and stabilizing selection, and we're going to talk about them in a lot of detail. And then we might mention antibiotic resistance if we get some time. We probably will leave that to next lesson now. Right, so selection pressures. There's some notes on the board, but I'll talk you through them. The point here is that variation levels pretty much never stay the same. Okay, the amount of variation in a species is constantly changing, so it's increasing because of new mutations, and it's decreasing because of random deaths, alleles being lost when people die, basically, and also because of natural selection. So I'm just giving you the answer to the question at the bottom of the page. That's quite terrible, isn't it? Never mind. There's almost always a selection pressure. Now, underneath that, then, can you write down? I've given you 3 examples. Can you write down examples of things which will lead to change in the levels of variation in a population? So we should have the top 2 paragraphs of notes, and then the question underneath. Just write down a list. I've given you 3 examples. There's a few. Right, so when we're thinking about things that will increase or decrease variation, the main thing that increases variation is mutation. That's pretty much the only thing. Things that will tend to change the amount of variation, natural selection, so some kind of change in the environment. You could have a competitor, you could have a predator, you could have mate selection. So it could be that suddenly the opposite sex starts to find something particularly attractive, that therefore will lead to more variation because we'll have different groups of individuals acting in different ways. You could have natural selection in general would include things also like artificial selection as a separate thing. It might be that humans deliberately decreasing or increasing the amount of variation. Now, we can kind of classify all of the things that will tend to change the amount of variation as something called a selection pressure. Now, a selection pressure is any factor that will tend to change the amount of variation within a species, a population. Okay, so normally, if you look at this graph on the, well, it says my right, I'm never quite sure if it's your left or not. There's a graph, you can see that's a normal curve. And by the word normal, I'm sure you know, I don't mean something that is usual, but that kind of up and down, we might also call a bell curve, is called a normal curve. And for most quantitative variation types, it will form a normal curve like that. So weight is an example. Most people are a middling weight, an average weight. Some people are extremely heavy. and some people are extremely light, but most people are in the middle. And so if you kind of count everybody up, you get a distribution like that called a normal curve. And selection pressures will change the normal curve. That's what they do. Sometimes they'll cause the normal curve to move in one direction, so maybe people in this case might get fatter or thinner. Sometimes they'll tend to change the width of the normal curve, so they might cause there to be more people on the outside extremities or less people. But in one way or another, a selection pressure is going to change the normal curve. Let's do an example of that. So in your head, don't need to write this in a book, what would be the relative survival frequencies of these weights? By which I mean, are they going to make you more likely to survive or less likely to survive? So massively underweight, I'm talking ridiculously skinny, massively overweight, similarly, or average weight. Which ones are going to be less good and which ones are going to be more good for survival? Have a think. So, hopefully everyone understands that massively overweight is not good for your health. You're going to have heart attacks, and you're going to have diabetes, you're going to have all kinds of horrible things, and that decreases your survival quite dramatically. Less well known, massively underweight also dramatically decreases your survival, almost as much actually, though not quite. Massively underweight causes a lot of issues. It depends on what it's caused by. If it's caused by a bad diet, then that causes all kinds of issues with bones, with growth, and we have to grow correctly, repair your cells, that kind of an idea. If it's to do with your metabolism, i.e. you're just burning up your fat too fast, for instance myself, I'm quite underweight because of my metabolism, actually that leads to increased risk of various cancers and all kinds of nasty things. So actually being massively underweight is not quite as bad, but it's almost as bad as being massively overweight. And an average weight is going to give you peak survival. So then the question is, if that is true, what should happen to my normal curve over time? If I give it 1,000,000 years, what should happen? And hopefully we've got the idea that the high extremity and the low extremity should be there less and less. There should be less and less of those people. Because if they survive less long, they're also going to have less children probably, and so less of their genes will be passed on. So if it's a genetic thing, then it will be less likely to be passed on. And what should happen is that the central portion, the average people, there should be more and more of the population at that level, until pretty much everyone is in the average. Now you might say to me, well, that isn't happening, weight is increasing dramatically, and of course there's an issue with environmental factors here, particularly with humans making bad choices, and the ability to get huge amounts of food. Yes, I agree, there are a lot more obese people than there used to be, but pure natural selection, if there was not for the NHS and people being able to live longer with these kind of things, then we should see what we described earlier, and that is called stabilizing selection. So, stabilizing selection is the first type of selection pressure we need to know. Some types of selection pressure lead to the average being selected for, average being best. And what should happen, as we said, is the curve should tend towards the intermediate area. So take down the notes there. Right. So, over time, what effect will stabilising selection have on the amount of variation between individuals is the first question. And what will this tend to do to the gene pool, to genetic variation, to the amount of alleles around? What do we think? And can you write down these questions in full sentences, please? So, hopefully you've understood that stabilising selection should decrease variation. Beforehand, there were some thin people, there were some fat people, and they were most in the middle. Now, if we give that 1,000,000,000 years, all the thin and fat people should just not be there anymore, we should all be average. So there's a lot less variation. That will decrease the gene pool. That will decrease the number of alleles around. You might say, well, that's a good thing, all the extremely fat and extremely thin alleles, they'll be gone. But of course, those thin and fat people will have had other genes which they tended to have, on the same chromosome, let's say, or just found in the same kinds of families, which will also be removed. So maybe there's some people who have a load of thin genes that made them very thin, but they also had a load of cancer resistance genes. Those will die out too, because the thinner people will tend to survive less long. And so losing members of the population because of selection causes decrease in the gene pool in bad ways as well as in good ways. If there is a good way to lose people, obviously there isn't, but you know what I mean, biologically. Fine. Now, the other we're going to learn about, so stabilizing selection is that one, that's what stabilizing selection is. The other we're going to learn about is directional selection. Some selection favours one extreme over the other. Now, I'm going to come across a bit harsh here, but in men, which of these is most favoured selectively? Being extremely small or being extremely tall? Which of those do you think is going to have the least bad effect or the most good effect? Sad to say, for extremely small people, it is true that being extremely tall is more favoured genetically than being extremely small. Because of various biases and just different levels of attraction people feel to other members of the species, more tall people tend to have more children than very small people. There are exceptions. But then what should happen to the normal curve over time? What do we think? Well, what should happen to the normal curve over time is, if that's true, if tall people have more children, we should get more and more tall alleles in the population, and so more and more tall people should exist. And therefore, what should happen to the normal curve, I hope you'll understand, is that this kind of thing should happen. This is in 1900. The average height was 155 to 160 centimetres. Nowadays, we're looking at more like 1.8 metres, something around 6 feet. So we've gone up by 20 centimetres in height in 100 years, 117 years was when this data was taken. And that is very dramatic. It's a very large increase. Right? And this is all down to two things. Firstly, better diet. That may actually be the main factor, but also down to selection pressures. And this is directional selection. And hopefully you can see the curve is the same width, it's the same height, there's the same kind of variation around the mean. The mean has just jumped to the right a long way. And so directional selection favours one extreme over the other, and so the normal curve just moves. And there's a kind of a set of notes there which summarises that. It causes the mean to change. I'll leave you 30 seconds to pause me to do that. Fine. So what should happen to levels of variation here is the final question I want to talk about with directional selection. Hopefully you've understood, levels of variation don't change. Mutations lead to more and more tall alleles, and selection leads to the removal of small alleles, but over time the width of the curve stays exactly the same. It's just that everyone jumps to the right. So variation is the same, we haven't changed the variation. We've removed some alleles, the small ones, and we've removed some alleles, so we've added some alleles, more tall ones, but the variation stays the same. Okay, there's a third type of selection pressure once we've had stabilizing and directional. This one is technically on paper 2. You will not be asked about this in paper 1. It will not be on your mock this year, but you do need to know it for the A level, and it's very related to these two. So I want to cover it now, and I would like you to watch the video and learn it, because it will help you a lot in year 13 when we come on to a similar topic, which we're going to do in year 13. And it will be a shame if some people know it and some people don't next year. So please don't close the video. And this one, I'm going to illustrate with salmon. Well, that is what salmon looked like in the wild. As you can see, a beautiful beast. In salmon, Females prefer larger mates. It's actually quite a common thing in the animal kingdom. I mean, less so in things like insects, but it tends to be the bigger the male, the better. I mean, they're better able to protect themselves. If you're hanging around with your mate, they're better able to protect you, but it also means that your offspring are more likely to survive because they're going to be bigger too. So they prefer larger mates. So what will tend to happen to the normal curve for salmon size over time is the question. What's going to happen is that the normal curve should move towards bigger males, just bigger fish in general. That would be what kind of selection? Stabilizing or directional? Directional selection. But there's a wrinkle here, there's an issue here. And the issue is that salmon don't have issue with rape. And there are some salmon who force females to mate with them in a very sneaky way. And what they do, if they are very small, they do what they call sneak mating. What they do is the female sees a large male in the vicinity. They kind of do a little mating ritual. The female releases her eggs, ready for the large male salmon to release his sperm into the water and fertilise the eggs. And what happens is the tiny little male salmon darts in, sprays his sperm all over the place, fertilises all the eggs, and then swims away as fast as possible. It's called sneak mating. It only works if you're very small and very fast. Because if you're big, the female will see you in the area and the male will chase you away. If you're small, you can hide in crevices nearby and you can be quite fast in sneaking. They are very successful. In fact, they're equally successful as the large salmon. So then what should happen to the curve? Now you've got large salmon being selected for and small salmon being selected for. What about the medium salmon? Well, now the medium salmon is actually the worst possible variety, mating-wise. You're not big enough to attract a female, or you're less likely to than a big one. You're not small enough to sneak, so you are going to have the least number of children because you are medium. So what should happen to the curve? Well, let me show you. Hopefully you're kind of trying to think about it already. You're going to get something that looks like this. And actually, this is called selection against the mean or disruptive selection, as you can see on the top right of this table. I'll bring it back just to show you the word disruptive selection. Fine. So disruptive selection is where the mean is the least good of all the options. In this case, because it's medium salmon and they can't sneak. Now, big, good, small, good, medium, bad. What happens then is you get a weird normal curve like this. And slowly over time, there'll be more and more and more very small alleles, so to create smaller and smaller and smaller sneak salmon. And there'll be more and more and more very big alleles to create very big salmon. Where those two are mixed up in the same individual, let's say you have one small and one big, you'll be medium, you'll die without having any children. And so what we should end up with is a population with a large number of small fish, a large number of large fish, and eventually, if you leave it for long enough, this can lead to speciation, which we're going to talk about next lesson. Excuse me. The creation of whole new species. Because what's going to happen eventually is there's not going to be any medium salmon anymore, and eventually the big salmon will just breed with other big salmon, the small salmon will just breed with other small salmon, because their alleles will be so different. But we'll come to that next lesson. Regardless, this is disruptive selection where the mean is the worst possible, and so you end up with two separate evolutionary strategies that might eventually lead to whole new species being created. So we've had three types of selection now, covering all the options. Stabilizing selection, where mean is best, and you tend towards the mean. Directional selection, where one of the extremes is best, and so you tend towards that extreme. And disruptive selection, where both extremes are best, but the middle is the worst, and therefore you will tend towards the kind of graph we'll see on the board here. Right, I'm going to give you a bit of a long task now. This will take you a while, unfortunately, because it's quite a big table. And what I'd like you to do is I'd like you to draw this table into your books, / pieces of paper, fill it in, and it should be a tick box exercise where the mean changes, well, tick which one you think it is. And I'll give you the answers. I anticipate with the amount of words and the size of the table, it'll take you 4 or 5 minutes. So pause me for that level of time you need and just have a go at it, okay? So, answers. The mean changes. Well, think about this carefully. Stabilising? Definitely not. In fact, the mean is the best, so we're just going to stay on that mean. Directional? Definitely, because we're going to move the whole curve along, either downwards or upwards, so the mean is going to shift. And then disruptive. Well, let's have a look at our graph. With these two curves, if you actually added them all up and divided by the number, the average would still be directly in the middle, because there's going to be about the same number of large and small fish. And so the mean actually doesn't change, it stays the same. So only stabilising. Only directional is the first one. Tick. One extreme is favoured over the mean. Directional. That's what directional is. The mean is selected for. Stabilising. That's what stabilising is. Changes the range of variation. Well, stabilising we said did, because you lose the extremes and everyone goes towards the middle, and so there's less variation. Directional we said didn't. The whole curve just shifts, but the variation levels stay the same. The range stays the same. Disruptive, yes, definitely. We should get more and more large fish and more small fish, or whatever our example is, and so the whole range shifts. Spreads out. Leads to distinct groups forming with different evolutionary strategies. That is disruptive and only disruptive. For the other two, there's just one best solution, so there's only one evolutionary strategy. Maintains one normal curve. That's stabilising and directional. The only one that doesn't is disruptive. It creates two separate normal curves. Leads to a concerted change in the mean in one direction. That would be directional. We're moving it in one direction, either up or down. And tends to favour a stable population. Well, the clue's in the name, stabilising. We're going to keep a stable population. Eventually, everybody should be the same. And it's worth noting there are literally millions of examples of stabilising selections in humans, which we all know about. We tend towards two eyes. Having one eye, being born with one eye, is going to make you less successful evolutionarily. Having 3 eyes is going to make you less successful evolutionarily. It's harsh, it's biased, but that is true. People find people with 2 eyes attractive. Same with 2 arms, 2 legs. Literally anything that's normal about a human has been selected for by stabilising selection. And every now and then there will be people born with extra limbs and extra appendages, but stabilising selection will lead to us all tending to have the same number. There's a lot of examples of directional selection. There are a lot of things where one thing is best, and so we all tend towards that. But disruptive selection, there are very, very few examples that you need to know about. In fact, there's none you need to know about. There are very few examples in general. So you'll tend to come across in your exams stabilising and directional pretty much being the ones you asked about. Particularly in paper 1, as I said, only stabilising and directional you'll be asked about. Fine. Now, we're going to finish off this lesson, and we've been talking for a little while. We're going to finish off this lesson with a little bit of work on bacteria. And this is a specific example that the A-level examiners do expect you to know. And this is about antibiotics. Now, have a look at the questions on the board. Now, this is from GCSE, and I think you also covered it in the cell biology topic of A-level, but I'm not sure because I didn't teach it. So have a look. So, the small circles of DNA that are found outside of the chromosome in prokaryotes, that would be a plasmid. A plasmid. The kind of information that these pieces of DNA have on them, hopefully we can all remember, it's not strictly necessary for survival, but it confers some advantage. Things like antibiotic resistance are the classic examples of things you might find on plasmids. But it could also be things like being able to adapt to particular increases in heat or pH, something that if you're in a normal environment you wouldn't need, but don't hurt you and might help you. So most of the time bacteria do not require antibiotic resistance. It's stored on their plasmids but not used. You spray some antibiotics on them, the ones that have it will survive, and so they keep these plasmids around. Fine. Now, we're just going to touch on it in literally one slide. If I put some bacteria on an agar plate and add penicillin, and after a week, 90% of the bacteria are dead, but 10% are left. Firstly, what do we call those bacteria? That's a fairly obvious question. And secondly, what they want you to be able to talk about is what type of selection is that? So, these bacteria are called antibiotic-resistant bacteria. And the type of selection, I hope you're aware, is directional selection. Now, there will be different levels of antibiotic resistance in different bacteria. Some will have none. Most of the ones that are left will have a small amount of antibiotic resistance, and some will have a lot of antibiotic resistance. By spraying the bacteria with penicillin, by adding penicillin to the bacteria, I have added a selection pressure, a new factor which is going to cause them to be selected in one way or another. In this case, all the ones that didn't have antibiotic resistance will die, all the ones that do will survive, and so if I have a normal curve of antibiotic resistance, the whole curve will shift towards the antibiotic resistant end of the spectrum. It's directional selection. Now we're going to talk about this a whole heap more on the next lesson, when I'm going to talk you through a practical we're going to do when you get back into school on antibiotic resistance. So be prepared for that next lesson, but that's all I wanted to say about this for today. So from today you should know, and I'm going to pull up the learning objectives from the beginning of the lesson. You need to know what is meant by directional and stabilizing selection. You need to identify types of selection from distribution curves. You need to interpret data relating to the effect of selection, so that's being able to look at those graphs and tell me what's happening. And then you need to apply knowledge of types of selection to explain antibiotic resistance, which we've just talked about, and human birth weights we covered during the lesson on stabilizing selection. That's it. If you can do those things, then you are well able to do this lesson. And then we added disruptive selection as a final option, which we'll cover in a lot more detail in year 13, but which would be helpful for you to know now. Have a great day, year 12. I look forward to seeing you whenever we're able.